This Week in Virology, the podcast about viruses, the kind that make you sick. It's time for This Week in Virology. This is episode 202. It's recorded on October 5th, 2012. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Today, we're on the road again. We're in Lincoln, Nebraska, at the University of Nebraska. We're at the 12th Annual Virology Symposium. We just heard 3 great talks. I'm not including mine.
Who is this?
I can't judge myself. You guys can do that. Well, there were 4 talks today. Now I thought it would be nice. There is a Center for Virology. It's called the Nebraska Center for Virology. And it's got over a dozen virologists, which is just great to have all these virologists in one place. So I thought we'd do a TWiV here and highlight some of the diversity in virology. So I've got 3 individuals up here who kind of represent the gamut of virology at the center. And let me introduce them to you so we can start talking. On my right, this is the fellow who actually suggested we do a TWiV here. He's a professor at the university and co-director of the Center for Virology, James Van Etten. Welcome to TWiV.
Thank you. Glad to be here.
Thanks for asking. I didn't think he knew about TWiV. And first he invited me to the symposium, and then he said, "You want to do your little podcast there?" I said, "Sure, it's a good idea." So thanks a lot. To his right, he's the director of the Nebraska Center for Virology, also a professor here, Charles Wood. Welcome.
Thank you. Good to be here. And thanks for coming here.
My pleasure. By the way, my first time ever in Nebraska is here.
Brought you great weather.
And last but not least, all the way on my right, also a professor here, Jack Morris. Welcome.
Thanks for attending. Looking forward to this.
You're a TWiV fan, aren't you?
I'm a TWiV fan. Yep.
And we also have a nice audience here, 250 people, would you say? And how many of you have heard of TWiV? Can you raise your hand?
Wow.
It's pretty good. Almost half, not quite half. Next time, it'll be 100%. I know every time we do these on the road, it gets bigger and bigger. So welcome to the recording. So as you know, in TWiV, we try and bring virology to everybody. I talked a little bit about TWiV earlier, and I forgot to mention that this is not just for virologists. We did a poll not too long ago of 1,000 people, and only 35% identified themselves as scientists. Okay, so the rest are non-scientists, all different sorts, but this is bringing virology to everybody. We even have high school students listening in on us. All right, so let's talk about viruses. That's what we do here. And first thing is to find out how everybody got here to this place. Let's start with you, Jack. What I know about you is that you're Canadian, right?
Yeah, so I did make that point a little bit earlier. But actually, I came here in, I think it was 70. About 1970 as a graduate student. I had been working at McGill, did a master's degree there, and I stumbled into doing plant virology, and the fellow that was doing plant virology there was from the University of Nebraska. He had worked with a couple of famous plant virologists, Ellen Ball and Myron Brackey. And so it was mainly for that reason that I ended up coming here to go to graduate school. And spent a couple of years here, and then did a couple of postdocs in Vancouver, and my first job at the University of New Brunswick, and then I got an opportunity to go to UC Berkeley, and I spent 15 years there, built a program in plant virology there. Then an opportunity came up to come back and be the director of biological sciences at Nebraska, and I said, I talked to my wife, and I said, "What do you think?" We liked it here. And so we came back.
How long have you been here now?
So it's about, since 1990, about 22 years.
All right.
And
throughout your career, you mainly worked on plant viruses.
worked on plant viruses. We worked on viroids and did some work on insect viruses for a number of years. So I've had a pretty diverse set of interests in different types of viruses, but mostly on structure and function of small RNA plant viruses and on plant defense systems. Plants have an immune system.
All right. We'll come back to you. Let's find out. Charlie and I have some history in common. Where are you from originally?
Well, I was from Hong Kong originally.
Yeah.
I spent most of my life in this country. And yeah, we kind of crossed paths a little bit.
You got your PhD at Columbia,
in Columbia,
right? Which is where I
yes.
am now. But we didn't overlap.
Not that
at
time,
all.
yes. I guess I was a student there in Columbia, and
With
Alvin Cabot, the late Alvin Cabot. So I was trained as an immunologist, not a virologist. But then, of course, at that time I hated virology, because Harry Ginsberg was there as the chair. And you know Harry Ginsberg, he's a strong arm person, adenovirus.
Harry hired me. Yeah, he
Yeah.
must have some. He has to have some good taste, right?
Yeah, he did, I think so. Well, at that time, I still wanted to do immunology after finishing PhD. So I ended up at MIT, working at Susumu Tonegawa, and we moved from Basel to MIT. So I was the last person that worked on immunoglobulin genes, if you remember. And then I decided to learn some virology because of a new disease called AIDS at that time. And so since then I became a virologist, but I really love it because it's really a cross talk between virology and immunology. Mm-hmm. It's really important.
Of course.
Yeah.
So you got your PhD at Columbia, which is where I am now, but I wasn't there at the time. And then you postdoced at MIT.
That's right.
We overlapped maybe a year at MIT. But now here in Nebraska, this wasn't your only position. You were elsewhere?
Well, I was a faculty at University of Kansas, so I loved the Midwest, and then a faculty at the University of Miami. Well, I learned of Nebraska because we fought those championship game, if you remember. And it was Nebraska-Miami, and Miami actually lost, so I decided to come to Nebraska.
It was the football.
It was football.
Yeah.
That was eventually. Yeah, well, I mean, Miami people love to come to Nebraska. We just recruited the athletic director, which is in Miami as well. So we have a lot of crosstalk and exchanges between Miami and Nebraska. And I've been here 16 years, so it's wonderful. Time just passed.
Wonderful. All right. And Jim, I know the least about your bag. Where are you from?
Well, and these guys are rookies in terms of being in Nebraska. I came in 1966. So anyhow, my background is that I came out of an antibiotics lab at the University of Illinois and graduated in 1965. And I was in the plant pathology department and was offered a position at the University of Nebraska at the time. But I had a fellowship from the National Science Foundation to go to Europe. And after visiting here, the net result was they agreed to hold a position for a year and a half. I came to work on fungi. And anyhow, I spent a year at the University of Pavia in Italy in the Department of Genetics and learning what this new field of molecular biology was all about. And at that time, it was basically looking at protein synthesis where you took a message called polyuridylic acid and made polyphenylalanine. You played around with ribosomes and all this stuff that really nobody knew much about. So then I came to Nebraska the following year, and the lab I was working in, I shared with a woman who had just started at Nebraska, whose husband was hired in the chemistry department. She was a microbiologist and was looking for a place to work, and we ended up sharing a lab, and that's relevant to the point I'll make in a minute. So anyhow, I spent the first 16 years here studying fungal spore germination, which was my background out of this antibiotics lab. When sharing the lab with Ann Vitiver, she discovered what turned out to be a very unique bacterial virus called Phi6, which was the first double-stranded RNA virus known, had a lipid envelope, had a lot of interesting properties. And so I got involved with this because of the lab sharing. So that was my introduction to virology. I've never had a course in virology or mycology. And then that led to about 1979 or 1980 when I was drinking beer with a colleague who was a former director of the School of Biological Sciences before Jack became the director. And he said something, I knew something, and we ended up doing an experiment which led to the discovery of a whole new field of giant viruses that infect algae. And these are viruses that have encode for over 400 proteins, transfer RNAs. Of course, we didn't know any of that at the time. But in the course of starting with these, I, over a 2-year period, dropped my other 2 projects, which were NIH supported, and starting in about 1981 or 2, then we spent the rest of our career studying these viruses that infect algae and have had all kinds of fun studying them. So I'm the one that doesn't know much about viruses outside of the few double-stranded RNA viruses, and then of course these giant viruses like pox viruses and now amoeba viruses, which are these things that have over 1,000 genes.
So you spent time in Italy.
Yes, I spent a year there as a postdoc. In fact, I have 3 collaborations with labs in Italy right now. And last summer I went to a meeting in Brussels, but spent the week before where I was 72 hours in Naples, 24 hours in Genoa, and 48 hours in Milan, all with people I collaborate on different projects with these viruses.
You speak any Italian?
Only the swear words. And that has gotten me into trouble once or twice, believe me.
I'd love to hear you speak Italian with that accent.
We'll do that off camera.
My son is taking Italian in high school, and he was visiting, I think, Boston University. He went into the bookstore and came out with a book of all the bad things you can say in Italian. It's this thick. It's an inch thick. It's unbelievable.
We could probably talk to one of each other.
So what was the little conversation that led to you working on these giant viruses?
Well, like most of my career, it's been by sheer luck, and in this case, we actually got involved for the wrong reason. So we had worked on the double-stranded RNA virus with Ann Vitiver. I worked with fungi, and fungi in the mid-60s and onward were finding these double-stranded RNA viruses as fungal viruses. And so I knew a lot about these. I always thought of algae as just green fungi. So when in this conversation, this colleague of mine said he had reason to think that he might have a virus on an alga. And there's a whole symbiosis story and a lot of other aspects I'm skipping over right now. But anyhow, I said, well, I know there's almost nothing known about viruses in algae, and maybe this will be another double-stranded RNA because I knew about these other double-stranded RNA stories. So the net result was that we took a look at these and over a couple of years found out we could grow them and plaque them, which was the day I made the decision to drop the other 2 projects and start working on these. But clearly we found out very shortly that these were huge DNA viruses and not what the reason I thought we might find initially. So really the thing that took off on us was the curiosity-driven research. We knew there was almost nothing known about viruses in this area. There's an interesting biology, which I have not mentioned. I'm glad to talk about it, but because of time I won't. And that's turned out because these things encode for over 400 genes, of which now about 40% are, there's a pretty good idea what they might do. But some of these proteins, of course, have never been found encoded by viruses before. This has led us into DNA restriction into nucleases encoded by viruses. We are the world's smallest potassium ion channel protein ever found that on anything, and we work with 2 electrophysiology groups in Europe, including one in Milan, Italy, and now we're doing a lot with sugar metabolism, which these viruses encode for a number of processes involved with sugar metabolism, including apparently encoding most, if not all, of the machinery to glycosylate their major capsid protein, which is clearly different from what any other virus does.
You sure they're viruses?
We get that question every once in a while because of all the genes. That yes, they do. Everything would indicate they're a virus.
Why are they so big? So is every algal virus that you find this big?
No. Well, for a long time, and we just work with three that we work with. These are ones that infect freshwater algae. But after we found these in the early to mid 90s, a few other people started looking at viruses in marine algae and finding large DNA viruses. And so for a while, it looked like the story might be that all viruses that infect algae might be these large double-stranded DNA genomes. But it's turned out in the last 5 or 6 years, as people start to look, there's been a double-stranded RNA virus found on a marine alga. There's been single-stranded RNA viruses and also single-stranded DNA viruses on algae. So it's clear that algae have the whole gamut of virus types. And it's just a matter of people looking for them.
So what algae, you said you're using freshwater algae. What are the names of them?
Okay, these are algae called chlorella, and the ones that we work with are normally symbionts in protozoan. And when the algae live in the symbiotic stage in the organism, they're resistant to virus infection. They're only inhibited by the virus, they're only infected by the virus once they're free of the protozoan. Now, fortunately, in the lab, we can grow these in culture, and so we just study the virus-algal relationship. But there's another aspect of this, and that is that we can go out in nature and at certain times of the year and just getting fresh water from a pond or a lake, and we have found as many as 100,000 infectious particles per mil of native water. And so there's it was a whole ecology that nobody knew about, and we're still struggling with in terms of trying to understand, but it's not even clear that the alga that we work with in the lab can even exist free in nature. So there may be another host out there that we don't know what it is, or host.
Well, what was the origin of the alga that you work with?
Well, it came from a protozoan called Paramecium berseria, and the fellow that I had the conversation with was actually studying the alga protozoan symbiosis system, and had just taken some EM pictures of the alga and happened to notice that something looked like a virus in that. And that was really the conversation that got this whole thing started.
So explain this. There's a symbiosis between the algae and the paramecium.
Yeah.
Who's inside who?
Okay, the alga is inside the paramecium.
Okay.
And what the experiment that they had done was to isolate algae. Actually, it was actually done with hydra, but we'll use paramecium as an example. They observed that if they isolated the alga from the paramecium and took electron micrographs of the alga over a period of time, they saw a buildup of things that looked like virus particles. And that was, in fact, he didn't even recognize it. There was another visitor that came through that happened to be looking at I didn't even know anything about this. Happened to look at his notebook and said, well, gee, that alga looks like it's got a virus in it. Went on his way, and that was then about 3 days later when I was talking to him. I was hosting either a visitor or a seminar speaker at my house. He just happened to be there, this fellow by the name of Russ Meitz, by the way. And we both remember exactly where we were standing in my kitchen. We don't remember the type of beer. I've had that question, but anyhow, it was that conversation that led to this whole thing.
So when the algae is inside the paramecium, it has virus in it, but it doesn't multiply. Is that the idea?
It turns out the virus, you can't see it there. I think it's because the the virus cannot get to it. I think it's a compartmental thing. But if you isolate the alga from that, then at the time we thought it might be lysogeny, but I think it was probably an external infection. I think these viruses are quite common. So if you squash the paramecium, the algae are released, then they're attacked by the virus. And that was what they'd taken pictures, 2 or 3 hours after they isolated the algae from the paramecium, and that had the particles.
So once the virus gets in, it kills the algae.
Yeah, this is a lytic.
They're all lytic infections.
Yeah. Well, all the ones we work with, now there's a a brown algal virus that's related to ours. It actually has a lysogenic life cycle. And that has a very interesting biology. And there's a question even with ours. We've gotten some very interesting plaques in recent days from some samples we just collected that are cloudy. And so there may be an aspect of this story yet to be developed.
So to make a plaque, the algae have to stay on the dishes, is that right?
Yeah, it's just like you would do with a bacterial virus, where you have a soft, you have an overlay on an agar surface with the alga. You mix this, when you make the algal surface, you mix it with various solutions of the virus. And then 2 or 3 days later, you get a beautiful plaque on a nice green lawn. Nice. Yeah, it's I can tell you a story about that. So when the first time I tried it in about 1982 or 3, I could see after a day, if you held the plate up, that there might be plaques. And I remember going around talking to janitors or anybody would talk to me about, man, this is exciting. And so that night, it was around Christmas, the School of Biological Sciences was having a Christmas party, so I drugged this plate. I had people that didn't know the difference between a frog and a virus, had to show them this plate that was starting to develop plaques. So yeah, that was an exciting time.
So because you can do plaques, you can do genetics?
You have the potential of doing real genetics, and one of the disadvantages is we still can't do molecular genetics on the system because we haven't been able to transform the host. And we've tried off and on, and we'll solve that problem, but that's been an ongoing project for about 30 years.
So you can't modify the genome and make directed
changes? Not
changes,
intentionally yet, no. That's the one negative. Yeah.
And algae are the only hosts that will replicate it?
Well, as I say, I've got reason to think there may be other hosts out there, but that's the only one we know of.
So
and they're highly specific.
If you put the virus on HeLa cells, will it do anything?
Now you're getting into something that's getting very interesting. The answer to that is open to investigation.
Because HeLa cells are easily transformable, as you know, so it's worth trying. Human cells, because I can imagine you jump into a pond with all these viruses and they're going up your nose. You get protozoal infections, parasitic infection, Naegleria, and that sort of thing. So I can imagine these big viruses getting up your nose as well.
Yeah.
But really, what I think, getting back to this question, I know they're not all big, but the big ones, they can code 600 proteins roughly?
Well, the ones we work with, the biggest ones encode about a little over 400, and they're related to this Mimi virus, which encodes for over 1,000 proteins. There's like 50 genes in common among them. And these viruses are also evolutionarily related to things like pox viruses, smallpox, or at least there's some pretty good reasons for thinking that, along with another group of viruses called the iridoviruses, which infect insects and some animals, and African swine fever virus, which is a quarantine virus on swine, which is a very dangerous virus if it gets into pigs. So these things, again, there's reason to think these may be very ancient viruses, at least the evolution. And some people have suggested they may go back to the time prokaryotes and eukaryotes actually split, came out of that same gene pool. There's some hand-waving with some of these arguments, but they're not totally ridiculous either.
So I know with the mimes, if you start with the 1.2 million base genome, you put it into a host and they begin to throw off DNA. And does that happen with your viruses, or
you mean DNA or virus? I mean,
so the mimes throw off genes. The genome reduces after a few cycles in.
Well, okay, I know that story. I actually reviewed the manuscript, so well, that's only under very, very, very specific situations where that happens. Yeah, and we do have some very big deletion mutants of our virus, so in that sense, there is a similarity.
But you see a stable genetic makeup in your viruses upon culture. They don't lose genes.
Well, right? For the most part. But now you can isolate some of these with large deletions. And this has always been fortuitously in our part. The paper you're talking about was that they passaged the virus through about 200 cycles. And at the end of it, they found some fairly good-sized deletion mutants. And again, what I don't know is whether, or I can't remember if that happened to the whole population or just a subpopulation.
Okay. Now, what are these doing in the environment besides, looking out for themselves and wanting to reproduce? Do we have any sense that they play a role in, limiting populations, carbon turnover, geothermal cycles?
In fact, they play a big role in the environment. All of you have heard of things like the red tides and brown tides, these massive algal blooms in the ocean. And you would have thought and some of these can disappear very rapidly. And you would have thought somebody 50 or 60 years ago would have thought that might be due to a virus infection. Well, it's only been in the last 15 years that people have found that the disappearance of some of these massive blooms is the viruses play a big role in it. And the ones that they've studied the most, again, are very closely related to the ones that we had discovered 30 years ago. So they have a huge role in terms of the environment. Most of you have heard of something called the White Cliffs of Dover, which occur off the coast of England. Most of that is calcium carbonate, which actually comes from an alga that is killed by these giant DNA viruses, and this has been going on for thousands, if not millions of years.
That's EHUX, right?
Yeah, EHUX virus. So yes, they have played a huge role. They do play a huge role in the environment, and of course, when you throw in all the bacteriophage, which are in the ocean, the biggest source of genes on the planet are viruses, without question.
Mm-hmm. So the EHUX is involved in cloud formation, and that's probably regulated by viral lysis.
That's right, yeah. In this case, you get a release of a sulfur compound from when the virus lyses the alga. This goes into the atmosphere, creates clouds, which leads to rain, yeah.
So if we are constrained for time, but if you could work on just one aspect of these big viruses and answer one question, one important question, what would it be?
Well, you're doing what the granting agencies want me to do. I've spread all over the map because we've had excellent collaborations with people who are experts in various genes. So, the one thing that does frustrate us is not being able to do genetic manipulations of the virus. And if we could transform the host, we think that would be a big way to get around that problem. But these things have so many interesting aspects. I don't want to be pinned down to one thing. At least I've made it, and we've met some wonderful people over the years in terms of collaboration, including one of the groups at Mount Sinai, a guy by the name of Mingming Zhao, who works on chromatin remodeling enzymes, and we have the smallest protein that methylates one of the histones, lysine 27. So we've worked with him. Somebody put up a slide today that showed Stu Schumann at Sloan Kettering. We've published a couple papers with him on a couple of our viral encoded enzymes. So yeah, we've had all kinds of fun.
How many groups are there working on these big viruses?
Well, in terms of the viruses themselves, very few. There's a group in Japan, but in terms of working on various aspects, we work with Michael Rossman's group at Purdue, who's a structural virologist. These viruses have turned out the structure to be much more interesting than what we originally thought. They were just going to be simple icosahedrons. Turns out they've got a unique vertex with a spike. They've got some fibers that extend from them. So in terms of groups we work with, probably at least 20 or 25 over the last 15 years on various aspects. But there's very few people that actually study the viruses as viruses. Now there's a couple of 3 groups that work on the EHUX virus that you mentioned earlier. But there's all kinds of room for people to come into this field. And where it's getting into a practical notion is that if there's this interest in using algae for biofuels, and there's a lot of good reasons for thinking of this, it's clear that if you're going to grow algae in large outdoor ponds, pathogens, including viruses, are going to be an issue that you have to deal with. And so there's a interest in that down the road, as well as using genetic components from the virus to genetically engineer algae.
Right. So that's why it's important for you to be able to modify the virus and introduce it into cells so you could do those kinds of experiments.
Well, and also then taking promoters from the virus to genetically engineer the algae to be resistant to various things. Yeah.
So you can easily make a case for doing this kind of research just for practical purposes, if not for purely educational purposes.
Yeah,
right?
we were fortunate. NIH supported us for about 25 years, I think mainly because we were sort of the odd person and people thought these were interesting. And right now, most of our funding is actually coming from some of these algal biofuels industry, but we still hope to get some more money from the National Institutes of Health.
So I will let you off the hook on that one question. If you agree to come back on TWiV another time, we'll do a whole episode devoted to your viruses.
Be glad to.
Is that a deal? Okay, because we'll have to move on now. Let's move over to Charlie Wood. I know you came back from Africa 2 days ago. It wasn't a vacation, right?
No, it wasn't a vacation. It was work.
What do you do in
Well,
Africa?
we have a research and training program over in Zambia, Africa. So I've been working there for the past 15 years. As expected, some of the infectious diseases they have there is really it's epidemic, like HIV. And of course, on top of HIV, of all kinds of cancers and strange diseases. So I've been working in over there just to look at transmission, specifically mother-to-child transmission, and how different environmental factors, other infectious diseases are affecting these transmission.
Of HIV?
Of HIV and other viruses. We work on the herpesvirus, which is known as the Kaposi sarcoma-associated herpesvirus. Mm-hmm. And these two viruses are really rampaging in that setting. So we have a team of about 20 people, laboratories, physicians, nurses, and data person to look at these two viruses. And it's amazing in terms of the disease burden over there. It changed my perspective of science, really. I've been a basic scientist looking at really laboratory stuff, molecular stuff. But going there, you're seeing the patients actually affected by these diseases. And you really go into more translational, how we can really, our research can be taken in the laboratory out to help some of these people. So really, I think it's amazing how we can adapt some science, what we know, into really in terms of affecting the population. So
you go and you guide people who work there to help you obtain the data that you're interested in.
That's correct, yeah.
You don't take a lot of people over on your own from here?
No, actually we try to build capacity. And it's very important that one of our goals is to make sure the locals benefit from what we are doing. Not only the outcome, but to build their ability to do the research. So we actually build clinics, laboratories, and we train people actually from Nebraska and then let them go back and they become our colleagues and collaborators, and so that they can really address the question locally. But at the same time, once we have the infrastructure there, people from Nebraska can go over there and also benefit from some of the things that they will learn locally. So it's really becoming a bi-directional exchange. And building capacity is so important so that we can do our own research.
So you were a basic scientist for many years. How did you transition into doing this as well?
Well, I think this is because of the disease that the virus we work on is HIV and this Kaposi's herpesvirus. The disease burden is so high there. And we want to see what we learn from the laboratory can be applied in the setting. Now, a simple example is, for example, we learn how to the transmission route. And at that time, when we started about 15 years ago, we don't know how this Kaposi's virus is transmitted, even the disease burden, how frequent it is in that setting. So by doing our research, finding out that there's very high prevalence of infection, and especially in children, they get an infection very early on, then we want to know where are they from, what is the route of transmission. So by learning some of these things, we can now actually design strategies. We now know from some of our work is that the virus is transmitted very early on via saliva.
From mother to child.
From mother to children, yeah. And then by the time the children get up to about 4 or 5 years old, they're almost at the adult level, which is about 40-50% in that setting. And on top of that, of course, HIV as well. So by understanding how they are transmitted, we can even implement public health strategies. Actually, we're doing that right now in our clinic. Our nurses are telling the mothers, well, you really should not have minimized saliva exchange with the children. So by this type of strategy, we can really impact, maybe reduce the viral transmission rate in that setting. So I think these type of things we can easily translate from what we learn from the laboratory into a real life setting. And this is I would call this implementation or a translational research from laboratory to the real life.
I would guess it's pretty hard to tell a mother not to kiss their kid, because that's basically how the saliva is transferred.
Yeah, it is not easy, but they listen, actually.
They do?
They do, because they see these children getting these Kaposis, they're getting skin lesions, and our nurses do a great job in terms of really educating them how to avoid doing it. And they're listening, actually, and they actually understand. We're talking about the people over there, they are not well off. I mean, they make like $50 a month in as a family, and they really have a very poor educational background, but they listen. I mean, I'm amazed that they actually remember what Kaposi sarcoma virus is from our clients. So it's really a whole different aspect of research.
So can you measure the effect of your interventions on the seroconversion or whatever it is that you're measuring?
That's some of the things we're following right now. And actually, we're trying to measure how much they retained our knowledge. And that's some of the things we're already measuring. And they do. They actually try to minimize the kissing, whatever.
So you said up to 50%. What are you measuring? Antibodies or virus?
Antibodies,
You
yes.
measure antibodies.
Antibodies.
So by a certain age, half of the population is seropositive.
That's
right?
correct, yeah.
And have you been able to lower that by your interventions?
Not yet, because we're working with a small population. So what we like to do is to take this model and then maybe take it to the next step to convince the local ministry, and then try to develop or implement some policy in the government. And then they can do some propaganda or whatever. I think that is when we see the impact.
Okay. So when the children are first infected by KSHV
Right.
at a very young age, is there any disease, overt disease?
No, that's the other thing. Like most other herpes viruses, normally you don't really see anything unless your immune system get messed up, like as in the case of HIV. So you get immunosuppressed, and that's the time when you see Kaposis coming up.
Right. So it's only when later on or whenever the children get
Right.
HIV do they get the skin lesions.
They do,
But
yes.
if you don't get AIDS, very rare to get skin lesions.
Very rare. But in Africa, because of the high infection rate, you do get so-called high incidence of so-called endemic Kaposis. These are normal, non-HIV infected people.
Right. So for comparison, what's the seroprevalence rate in the U.S., say?
It is probably around 5%. It's very, very low. So that is just also very interesting. The geographical distribution is not even throughout the world. Certain population is higher and other populations are lower. Now, you probably remember these so-called classical Kaposi when first discovered by Maurice Kaposi many years ago. They found that in the Mediterranean area,
Right.
These are the Jewish descent. So there are some of these genetic factors we still don't understand. Why certain population is high and others are low?
Are the viruses different?
We don't think so. Now, of course, there might be different minor subspecies, but they're not that different from each other.
So you study just mother to child, or do you look at the father, where there isn't a father usually?
They do. They do have fathers and actually extended families as well. And so one of our projects is to look at what are the sources besides the mothers. And we actually found out that the sources is not limited from the mothers, but could be from the household or even outside the household. Because children play with other children. I mean, they exchange candies, sweets, and they bite each other. So they have saliva exchange. And so we do actually trace them because we do genetic analysis of the virus. There are some sequences that they do vary, so we track them. So where are they from? So we have evidence that they actually could be from outside the household. And so it's not really restricted to the same family.
Yeah, sure. So if you didn't have an AIDS problem, you wouldn't worry about KSHV, right?
Probably not as important as what we're seeing right now.
Okay, so you also look at HIV prevalence in the same population?
That's correct. We also look at the HIV transmission from the mother to children.
Okay.
More or less on the virological sides. Basically, are there specific type of viruses that are transmitted versus those that are not transmitted? Are there any differences virologically?
How do you do that?
Well, we look at the bottleneck of transmission because, as you know, that HIV is a quasi-species, meaning that in every single infected individuals, there are thousands of these subspecies or genotype presence. But the thing that it's really interesting is when it goes from a host to a new host, you always have this bottleneck. So it's a filter. So from these thousands of species, it's going to filter down to maybe one or two minor species that goes through the new host. It happened not only in perinatal transmission, but happened to adult sexual transmission as well. So understanding how this bottleneck occurred has a lot of implication on prevention. So we're trying to look at what is causing this bottleneck from mother to child transmission. So we look at some viruses, and we believe that there are certain characteristics for those viruses that are transmitted versus those that are not. And so that's what we're going after in terms of the HIV side.
So you look at the species that are in the mothers and compare them to what's transmitted.
That's correct.
And that you do by sequencing?
By sequencing, genetic analysis. And also we do some assays in the laboratory to look at the biological properties such as the replication kinetics, the infectivity, binding, fusion, all those basic things, look at how the virus enters the host cell.
So when it's transmitted from mother to child, it's perinatally?
That's correct.
Is that correct? Because the mother's infected and then it crosses the placenta?
It can go different routes, either in utero transmission through the placenta or actually intrauterine, which is during delivery. That's the most frequent transmission route. And then, of course, after delivery, you can get it from breast milk as well.
So during delivery, so there's virus present at birth and it's contaminating the baby as it's being born. So how it will enter through cuts, for example, or the umbilicus? Do we know how it gets in?
Well, the exact route is still not clear, but most likely, probably, maybe ingesting some of the body fluid from the mothers, and get into the gut, and that time I think the baby system is completely not developed.
Yeah.
So that may be a route. I don't exactly how it gets in the baby is still not clear.
Okay.
After all these years and people studying.
So what is the seroprevalence of HIV in the same population that you're?
Well, in the beginning when we started 15 years ago, it was about 30%. Talking about 3 in 10 people. Now with the government education, the scale up of the antiretroviral treatment, people are being tested more openly now. The whole atmosphere is changing. So we're seeing a stabilization of the HIV epidemic in Africa, throughout Africa. So in Zambia, I think the official prevalence is about 16 to 18%, and it's maintaining stable. It may be dropping. But of course, there are pockets of areas that are much higher. And we see much higher in the population in the hospital.
So you said in the case of KSHV, you try to counsel the mother to reduce transmission, right? What do you do with HIV?
Well, in HIV, we work with the local government policies and trying to do counseling of every clients that come in, and also encourage them to be tested. I think the way to control HIV, as you know now, is to really early detection and try to prevent transmission. And I think it's a lot of hope recently because we have different strategies to really reduce transmission. For example, prophylaxis, treating infected people with antiretrovirus drug early that will reduce the viral load so that the chances of passing it on to the recipient is much less. So we are hopeful that if we can catch every infected individuals and treat them, you will probably eventually reduce the transmission rate, and then the seroprevalence is going to go down. So this really involve massive educational program.
But triple therapy is available?
It is available now in Africa, and because we have this government program by the U.S. government investing $15 billion into the PEPFAR program. And it really is helping in terms of providing drug to the people. So there is hope. I think that is really important. So people coming out to be tested. Before, I can see it, people don't even care because it's the death, it's a death penalty, basically. So there's no treatment. So the attitude, I can see a completely changed attitude. It's amazing that with people having a hope now, they are actually very adherent to the treatment program.
That's surprising because it's not without side effects. And here in the U.S., many people stop because it's not fun. So I would be surprised if you got good adherence there as well.
They do. I mean, but they still have side effects. So a lot of people cannot take it.
Yeah.
So those that can take it will adhere to it, but the side effect is still there.
So there's also been some trials of prophylaxis using.
That's correct.
Triple therapy, is that right, in prophylaxis?
They're using a single therapy, tenofovir, so far, but there are other drugs they are looking into. And it's actually quite effective in preventing transmission.
So what do you think that will be implemented in where you're studying these individuals?
I think so. Actually, the government is trying, at least in Zambia, the government is trying to they're trying to implement a policy to treat everybody that are found positive. So that will definitely lower the transmission rate. But our concern is it really sustainable? Because of all these outside aid to Africa, but if all these outside aid are stopped, can the government really sustain this treatment program? It's a big concern. So the African countries have to take ownership to be able to really take over. I think that's the hope we all.
Do you think that we need a vaccine to get around this problem?
Yes, we do, but I think if you remember, I mean, when Reagan was president, when the virus was first discovered in 1983, said we're going to have a vaccine in 5. Margaret Heckler, who was the Secretary of Human Health and Services, and she said we'll have a vaccine in 5 years. That was almost 25, 30 years ago. So it is going to be a tough one. Vaccine is going to be a little bit far ahead yet.
But the drugs alone are not going to be enough to really dent the spread, right?
It will, if they can treat everyone that is infected. Theoretically, you're going to keep reducing the infected people or transmission. It's going to go down. Yeah, there are models people can study that you can really get rid of HIV transmission.
Before we leave you, why don't you tell us a little bit about this very unique place, the Center for Virology? How did this come to be, and why do you have people doing the same thing in the same place?
Well, it's very important to have the environment or the critical mass for anything. And we're fortunate that even before I came, we have these great virologists here sitting next to me right here, and plus other in the audience. And we already have a cadre of virologists, but we are so scattered into many different departments. So opportunity came in the year 2000 when we had a chance to compete for a Center grant through the NCR, at that time, NCR IDEA program. So we were able to pull all the virologists together and put a proposal in, and we're lucky we got funded. So we were one of 18 centers back in 2000 that was funded, and here we are. I mean, 12 years later, we're still here, and it's a great thing because we actually continue to build on it, and we were able to recruit more virologists into different departments, in different areas, different viruses. We want to maintain the diverse expertise into plant, animal, and human viruses. And we also lucky to have
2 positions.
We're going to have more positions coming in. and we also are unique in a sense. We have a building, which hopefully you have a chance to see later on, that we all co-localized. And as our Vice Chancellor, Dr. Paul mentioned earlier, and it's quite unique in a sense. We were taking people away from the home departments, and we co-localized. So it's a lot of so-called, I would say, water we have to tread through, because it's never been done at the university, but we have great support from our administrators, from Chancellor down. And really, I think because of the successes of all the faculties we have in the center, and really able to maintain it. And we also are fortunate, as you heard, that we're going to be expanding. Our building is like 4 years old, and we had the opportunity to get this ARRA funding to increase our capacity by 30%. And Chancellor is now investing into growing human faculty number. So we already have 2 positions given to us this year to start expanding our center. And with the new facility, we're very hopeful that we're going to build it up to the next level. And we like to let the world know that we have great virology here, and we want people to come to Nebraska.
So have you started recruiting for these positions yet?
We are starting, and then we're talking to colleagues to see who are interested. And we really want to hire people that are at different levels, and especially senior people that can bring in a new perspective and take over the center and build it up to the next level. Okay. That's what we're hoping.
So if some of our listeners are interested, should they go to the website for the center?
Well, I think you just send us email. We want to really...
Send Charlie an email.
Or to any one of us an email. Send the vice chancellors an email.
Send them to Charlie.
He needs more email. People have gotten jobs through TWiV before, so you
okay, that's
have
great.
lots of very connected and interested listeners. So
yeah,
that's perfect.
So you have 2 positions.
Yeah, the plant virology one was just released this week, and and it hasn't even been formed as in terms of an announcement, but the money has been released to recruit for it. And then the more senior person, which was released 3 weeks ago. So those are tenure track positions.
And the senior person can be in any area of virology?
Can be any area of virology. We're open. And as a matter of fact, I think if we have the right person on board, I'm sure we can twist our vice chancellor's arm and our chancellor's arm to get more position to supplement that senior hire.
There you go. It's great to have a lot of virologists in one place. I'm very jealous, right, Terry, to have over a dozen. So I have 2 virologists in Columbia at the moment. I went there because Harry Ginsberg had collected 5 many years ago, but now they're all gone. And once you go below a critical mass, it's very hard to build back up again. And there's nothing like having people who think similarly to you. They really, really do help. So I think it's great that you guys have this. I hope you'll appreciate it, those of you that are here, and I hope it keeps on growing. That brings us to you, Jack. You've been a plant virologist all your career.
Well, plants feed us, and one of the major
they feed us.
Right. And so certainly one of the justifications for hiring plant virologists and doing plant virology has been the implications on food yield, which are significant actually worldwide. And I think one of the interesting observations I'll make as part of the field of plant virology, and Dr. Ding talked about it earlier, we can actually control virus diseases with fairly simple genetic engineering approaches.
So can you first just give us an overview of what kind of plant diseases are agriculturally important?
Pick a crop, and there will be 5 or 6 different viruses that you can find associated with that crop. Many are insect vectored, many are a consequence of human domestication and vegetative propagation methods. And so a component of the field is really focused on controlling and managing virus diseases in crops.
Tomato, corn, cucumber, peppers, anything.
We had a project not too long ago that with some facility we're able to genetically engineer soybeans to all 3 of the viruses that occur most commonly in soybean. Mm-hmm. So there are approaches now that allow us to begin to think about managing virus diseases. I think, though, one of the more interesting areas, we'll go back a little bit, I don't know how much time we got left, but one is, for a new plant virologist coming into the field, I think a strategic focus on virus discovery can lead to some really fascinating, I think, new observations about viruses. In fact, that's how my career started. I inherited a lab at Berkeley that was the electron microscopist lab, Robley Williams. And my discovery happened by looking in the refrigerator. And the viruses that I built a career on were those viruses that I pulled out of the refrigerator that he had used for his electron microscopy. And most of them were small RNA viruses, quite different from what Jim was working on. These are viruses that had 3 or 4 genes. I said, oh, that's the ones to work on, because you'll figure those out in a lifetime, right? And as it turns out, that really isn't what happened. What happened was they became really excellent models for recruiting students to do good projects. And probably one of the most rewarding things that I can say about being a virologist in the field was having smart students that wanted to do something interesting. And we had a couple. So we had all these small RNA viruses, and that was the pre-molecular biology era. And I had a couple of students came into the lab. Jim Carrington was one. And he said, we ought to do some molecular biology. And so I said to him, well, how are we going to do that? And he says, well, there's a guy over there in biochemistry, Bob, by the name of Bob Tejan. And he's doing some molecular biology. He says, I'm going to go work for him for a while. So I called up Bob and I said, yeah, how about teaching us how to do that? And there were a couple of students, Mark Young and Jim Carrington and Brad Hillman, that came out of my lab that learned how to do this kind of stuff quite early. So those small RNA viruses became model systems, tomato bushy stunt, turnip crinkle, as our friend Ding will attest to. They led to a lot of fundamental discoveries about things like RNA silencing, RNA virus assembly, and those are all really basic and fundamental studies that one can do, and at the same time carry a program that allows you to address the practical problems. One of the outcomes of doing our research in the development of molecular probes was the development of diagnostic tools for the strawberry industry in California, which actually paid patent fees that were some of the highest for the University of California system, built on diagnosing virus diseases and creating clean stock. So we do that now using these techniques for things like grapes and strawberries and a lot of the fruit crops now. So there's a lot of opportunities, I think, in the plant virology field, not only in understanding basic mechanisms of how viruses cause disease, which is where my focus is now, where most people don't really fully appreciate that plants have defense systems. They have an innate immune response, and that's another whole area of research that you can get involved in. And we heard a little bit about that today. Maybe an RNA-based response, but there is an innate immune response.
Well, in fact, this is only the second time we've talked about plant viruses on TWiV. The first time was in Brazil about a year ago. You may know some of the plant virologists.
And at ASV. You had a little, we did a little session.
Marilyn Rusink, that's right.
That's right.
So it's the third time. So we don't talk about it all that much, and we need to do it more. Mm-hmm. And in fact, you should come back and do a whole episode about it, because we don't have time to really get into it here. But I think most people don't realize that some of the things that you've said, the RNAi advances made in plants, and also the fact that it's agriculturally very important, and things are getting done right now. I think a lot of people don't realize that.
That's right. And some of the first structural models, the first crystallography were done with the virus system that I studied for many years, and by mapping the crystal structure that Steve Harrison had done with the molecular biology that we were doing, we were able to really get at and make mutants that had structural sense.
In fact, it was the first virus structure determined, right? Tomato bushy stunt.
Tomato bushy stunt, second to TMV, I think, yeah.
So as a way of, let's say, closing, I asked you a question the other day, and maybe we can revisit it. Someone had sent us a question to TWiV. Why is it that there aren't any DNA viruses of plants? And although it's not exactly true, you know what they meant.
Right, exactly.
They seem to be mostly RNA viruses. So what are your thoughts about that?
Well, much of our discovery, and we haven't really done discovery at the level of ecosystems in plants. So I asked my colleague Dave Dunnigan, I think David's here, because he has this diagram that says, well, where are the viruses? And yes, what we haven't found in higher plants are DNA viruses of the polyoma sort. There's no fundamental reason why we shouldn't find them that we can think of. Then again, if you look at that branch that is the planta branch of the tree of life,
Right.
there are all sorts of double-stranded DNA viruses of humongous size in the algae and so on. So these are all related. So why don't we have viruses of that sort in plants? I think there's probably two reasons. We either haven't looked properly, or it's some sort of an evolutionary black hole. And if there were DNA viruses, they somehow disappeared.
So you said last night that if they're too big, they might not be able to move within the plant. Is that another possibility?
Probably not likely, because what you got pox viruses in, well, not really pox type, but you got rhabdoviruses in plants. They're pretty big. You certainly have viruses of the size of papillomas in plants. They're pretty big. So plants have, or plant viruses have evolved mechanisms for passing through the cell walls that restrict movement. And so I don't really think it would be a movement issue. It might have something to do with the fact that plant meristems function a bit differently from rapidly dividing cells that maybe DNA viruses need in vertebrates.
So as you know, people are looking like crazy for all sorts of viruses in different environments, in oceans, in fresh water, in animals. Are people doing this in plants, or maybe it sounds like not?
There have been some ecosystem-wide studies, actually, to look at viruses in natural ecosystems.
Right.
And there, as a consequence, have been new discoveries of different kinds of viruses that aren't currently in the database. So I think that the answer to that is, now that we have the kinds of tools that allow us to do virus exploration and virus discovery that are different from the ones that we had 30 years ago, I think we're going to find all sorts of new things. We just haven't looked very hard. And if you look at that tree of life, how many branches have we explored? Very few. Animals and plants, right? But and some algae, and so a few other things.
Yeah.
But for the most part, we haven't really applied the modern tools of virus discovery to really looking at where viruses reside in our ecosystem.
Is that something you would like to do?
Oh, I think that'd be fantastic. That'd be fascinating. Except you got to learn how to do this bioinformatics stuff. So molecular biology was easy.
Well, you can you hire the bioinformatics people.
Yeah, that's certainly one of the things we have to think about doing.
On the other hand, viroids are only in plants, right?
As far as we know, other than the Hep B associated circle. So
why is that? We haven't found them yet?
Well, Shui can tell us why. We haven't really looked. And he's developed, we didn't get to that part in his talk, a bioinformatics approach for searching for small RNAs that are circular.
Yeah. So we just haven't looked. So there's plenty of opportunities in virology. But I don't need to tell this audience that, right?
I think so.
They're already converted.
Hope we don't.
All of you, everybody here is into virology, is that right? Yeah. Anyone not into virology here? No. That's good. All right. So usually in TWiV, we do email, but I'm going to skip that because we have a poster session that's starting soon, and we don't want to impinge on that because that's really important. But I would like to do our picks of the week. I have one, and I have some listener picks. And if you guys, Jack, did you want to pick something?
I know Charlie's got one because I was making him look on his iPad.
I know.
For something interesting.
I have to download this article. I mean, a lot of people have been talking about it. This is a new rhabdovirus that causes hemorrhagic fever in Congo, in Central Africa, just been published in PLOS Pathogen. It's fascinating because they just discovered in several patients, and 2 of them died. And one of them actually got antibodies and recovered. There was a nurse taking care of those two to patients. So whether this is an Ebola-like virus or not, it's something new. Because as a matter of fact, it's only 34% homologous to retrovirus, so it might be something different. And again, I want to point out that the reason they can piece this together real fast is because of this high throughput sequencing. They were using this Illumina just to get them and piece them together. So really, I think it represents viral discovery, represents high technology, and represents a new agent. And so I think it's a fascinating paper. It's easy read, so everybody should take a look at it.
I think they said it's the first time they could put together by deep sequencing of clinical specimens.
That's correct.
The whole genome. So they did it from serum.
From serum, yes. Yeah.
And I think there were 1 million copies of viral RNA per mil, which is not terribly high, so that's pretty good.
Yeah.
But we tend to gravitate to illness and look there, but as we know, there's plenty of viruses out there that don't make you sick that are really worth discovering, and there's a lot more to do. So my pick actually comes from my older son, and he hangs out on Reddit all the time. You guys waste time on Reddit probably too? No? It's because you're mature. So he's only 18. Anyway, he sent me last night a post he found on Reddit, which is bacteriophage graffiti. So someone found on the wall somewhere next to someone's apartment a drawing of a bacteriophage. And so I have an image of that, which I think is good because graffiti is usually pretty weird stuff, but a phage, so someone's a budding virologist out there. Maybe it's one of you guys who put the graffiti up there, I don't know. So phage graffiti on Reddit, that's my pick. I have two listener picks. These both came in last night. In case you don't know, those of you who don't know TWiV, we also have a page on Facebook. It's facebook.com/thisweekinvirology, all one page. We have 2,800 likes. Let's see if Nebraska can push it over 3,000. Okay, so you should just go and like the page because I know you're on Facebook. Anyway, last night Bob wrote, "Hey Vince, one of my past mentors did this piece. Thought you and perhaps the greater virology community at large would appreciate it." So this, I don't have internet here. Let's see if I can find. Okay, this is in the American Society of Tropical Medicine and Hygiene Journal. It's their blog, actually. It's an article by Dan Bausch, and it's called, it's about Penny Pineo. Does anyone know what that name is? Penny Pineo. You older guys are the only ones that would know. She was, sorry? Immunologist? No, she was a nurse, and she was among the first people to get Lassa fever in 1969 in Nigeria. She just died in August. She was 95 years old. So her story is so cool. So there were some cases of Lassa in Nigeria. She was a nurse. She took care of them. She got sick. They airlifted her to Columbia Presbyterian, which is where I am, but this was before I was there. They just put her in an airplane in the passenger compartment and shipped her back, okay? With no precautions. She recovered. They put her in intensive care. She recovered. They sent some of her serum to Yale. At the time, the Yale Arbovirus unit was there, and Jordi Casals began to work on, and he infected himself, and he got really sick. And they used her serum. They gave her serum to him. She had antibodies, and it saved his life, basically. And she's alive. She was alive up until August, and she knew this story. So this is a cool little blog about her. And you know, someone about a year ago emailed us on TWiV and said, you ought to interview Penny Pineo because she's just been discovered in Florida, and you should interview her before she dies. So I never got around to it. So anyway, this is a cool article on her. ASTMH remembers Penny Pineo, a pioneer in combating Lassa fever, 1917 to 2012. So thanks for that, Bob. And then last night also, a few minutes later, Janet from Heidelberg, one of our fans, said, don't know if you TWiVers have already seen this, but it's pretty funny and makes the point. It's a Penn and Teller little skit on YouTube about vaccination. Have you seen this? Okay, it's really, I mean, it's full of cursing, Penn and Teller, right? But it makes the point of why you should vaccinate your kids in a very illustrative way, basically. So check that out. We'll put a link to that in the show notes. It's really well done. So those are our 2 listener picks of the week. You can find this show, we're going to be posting this as usual, it's being recorded at TWiV.tv, that's our website. We're also on iTunes at the Zune Marketplace, and we also have an iPhone or an Android app that you can use to listen to these. You can find that at microbeworld.org. And we always answer questions and comments. We didn't have time to do that today, but we have tons of them. We love to get them. You can send them to TWiV at TWiV.tv. And I want to thank everybody for participating today. Jack Morris here at the university, thanks a lot.
Thank you.
Come back
Been fun.
again and we'll get into it in more depth. How's that?
That'd be great.
Would that be a deal? Thank you so much. Charles Wood, thank you.
Well, thank you. My
Thank you for
pleasure.
having us here and for doing this.
It's great to have you here. Welcome back.
Thank you.
And we need to get you a big red souvenir of some sort, right?
Oh, you mean a red sweater like
Something like that. With a big N on it.
you're doing? What is the motto here? Big Red or
Yeah, go Big Red.
Go Big Red? You know, I went to Cornell. That was Big Red. It's not in Nebraska.
Right.
Isn't Cornell Big Red? So what do we have, a conflict here? You know, Columbia is a center of football power, Columbia University. We once lost 150 games in a row. We were proud of that. I remember Michael Lai. Where was Michael Lai? USC?
Yes.
He came to give a seminar once, and his losing streak was even longer than Columbia's. That was great. Jim Van Etten, thank you for joining us.
Well, thanks for having me on your show, and I'm sure glad you came to Lincoln.
You come back again, right? Because we want to hear more about big viruses.
As I get old, I have plenty of stories to tell.
Excellent. We've got to get more of them. I'm Vincent Racaniello. You can find me at my website, which is virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.